The pleasure to introduce our next speaker, Rostislav Stoika. I don't know whether I pronounced this way. Okay, thank you. And I already told him I'm really surprised and happy to see especially this slide as an intro, because the football I have seen several times in Anna's presentations. So there is already a very sportive connection. Please.
Okay, good morning everybody. I would like to thank the organizers for inviting me. It's a great honor, and I would like to thank our German colleagues who are brave enough to come to Ukraine. They should really love Ukraine, hopefully.
We do.
They do. Okay,
we do
good.
already.
So I'll be talking about the nanomedicines and design and activity. Hopefully after my lecture, young scientists who are on the far rows will decide which specific nanomaterials they would like to produce. So how to create your own nanomedicine. And this is slide, you know it. So let me introduce myself. My main job is head department of cell proliferation and apoptosis at the Institute of Cell Biology, but I'm also working at two universities, at the Department of Biochemistry and of Biology of Danilo Galitsky Medical University. And this is nice city of Lviv. It's the city of lion. It was named to the lion, the son of King Danilo, who was there in Lviv, founded in 1250s. And it's a good location of our institute somewhere here. So we are in the center of the city, not far from the center. So our location is very good. And I can see this picture from the windows of my kitchen at the apartment. So the outline of my lecture will discuss several issues, statistics, because it's really very interesting, these statistics on the nanomaterials and nanotechnologies, general characteristics of nanomaterials for biology and medicine, nanoscale carriers for smart medicine, theory and literature data. Then 50%, more than 50% of my presentation is examples from our study of nanoscale carriers for smart medicines, and a couple slides on new trends in the development of biomedical nanomaterials and nanotechnologies. So I have some limitations in my lecture, so I'll be talking about true nanomaterials. It's not only the size, but it's because these materials gain new properties due to their nanoscience. If it's simply a nanomaterial, I'll be not talking about, but it's true nanomaterials. So which new properties? Improved water solubility, higher stability, prolonged action, capability of circumventing biological barriers, addressed action, decreased genotoxicity, and some others. So these new properties are gained in true nanomaterials. And I'll be talking only about the biomedical nanomaterials, since I'm a biologist and biochemist on application. So some statistics, just for fun, and it will be interesting for you also to know. This is 5 years old statistics, and we now have 43 types of nanomaterials, almost 2 million articles, more than 200,000 patents, and more than 300 products. And counting the areas of research, I was surprised chemistry is number 1, 31, material science number 2, science technology number 3, physics number 4, engineering number 5, and only number 6 is pharmacology, then biochemistry, molecular biology, biotechnology for microbiology. So mostly budget of the nanomaterials is from these natural sciences. And it's also interesting that leading organizations in nanotechnology, so those who are doing most projects, this is Chinese Academy of Science, then MTI, USA, Harvard University, USA, Georgia Institute of Technology, USA, Singapore, and other institutions. And funding, again, number 1 is National Natural Science Foundation of China, then National Science Foundation USA, National Institutes of Health, National Basic Research Program of China, and only on the 5th position is the European Union. So we should work to be higher in this list. And another interesting example, 2 years ago, the citations from researches of Chinese scientists is changed with the citation of the scientists from the United Kingdom. So again, some statistics, nanomaterials, and I'll be talking about these issues, nanomaterials for drug delivery, a little bit for gene delivery, a little bit for bioimaging, and a little bit self-assembled nanomaterials, because this is our future, self-assembled nanomaterials. So these are the smartest nanomaterials. And the last statistical slide, nanoparticles are the most widely used nanotechnological products, more than 400,000, and patents, more than 50,000 patents, and products, 12,500 patents. Then graphene, then nanofibers. So, second part of my talk, nanomaterials and nanotechnologies for biology and medicine. So I cited this from somewhere. It's B cells, vesicles, dendritic polymers, nanotubes, nanocapsules, nanospheres. So these are all pharmaceutical carriers for medicine. And there is a term, Smart Drug Delivery System, DDS, Smart DDS. So what it means? It means that there is a pathological locus in the body, so this information could be reflected in changes of pH, enzymatic activity, levels, glucose level, etc., etc., etc. It's responded by nanoparticles in cells, polymers, liposomes, and under the action of exogenous stimuli, temperature, magnetic, acoustic light, etc., etc. Delivery systems get drug release, and it's a treatment process in the body. So, stimuli responsive or smart or intelligent polymers. Different terms are used. Probably number 1 is smart. Then these two terms are also used for nominating these materials, mostly polymers. So they are such materials which are able to modify their physicochemical properties in response to variation of environmental factors. So these are stimuli response systems. And which environmental factors? Micro-environmental factors. It's temperature, ionic strength, pH, electric, magnetic field, and light. Magnetic field is external, of course, and light also. So the polymers of that type can respond to stimuli by altering shape, solubility, wettability, color, conductivity, light transmitting ability, surface characteristics. And the degree of this response can be treated as a change in intensity of applied stimuli. So pH, I will not stop in detail on these stimuli responsive materials. So they can be pH responsive. It means that at specific pH the drug is released. So can be pH of the stomach, and can be pH of the intestine tract. Then closer to what we are doing, it's a redox triggered drug delivery. And it is known that tumor cells have on different calculations from 10 to 100 and even more higher concentration of glutathione. They protect themselves in such a way from the attack of reactive oxygen species, and nano biotechnologies use that property, preparing a smart drug which is released due to presence of high glutathione. So disulfide bonds, covalent link, and the drug is released in such a situation only in tumor cells, not in normal cells. Enzyme responsive drug delivery systems, I will not stop, but they exist. Temperature, oh, sorry. Temperature responsive DBS. They are widely used because we can increase with electromagnetic field or laser the temperature in some locations, and this temperature can be elevated due to inflammation and some other pathological processes, and only in that specific site the drug will be released. Light, magnetic, ultrasonic responsive DBS, we were working with this due to our collaboration with Checkpoints. And a few words about self-assembly or self-organization. As I told you, this is the future of nanomedicine regarding drug delivery. Because one of the main problems, I will be talking later about that, is a non-targeted action, non-addressed action, non-specific action. And these systems can be self-organized in a specific place due to local interactions with the components themselves, and also with the components which are on the surface of the specific pathological cell. So for that, it should exist so-called adapter and linker, we have an example of such interactions, so-called scaffold proteins, during different sorts of regulations. So these scaffold proteins can form a complex called signals, and these signals can be very different, but all of them are very complicated. So this is the signaling adapter proteins for cell proliferation signaling. I will not know specific proteins, you can find them. This is for cell differentiation. So many proteins should get together, and in that situation, the signal for cell differentiation will appear. And very, very complicated signaling for cell adhesion. and see how many different proteins are participating in such a scaffold system or signalosome or cell adhesion signal. And of course, the interaction should occur, and such interactions are called interactome. And you can see there are clusters. Some proteins are interacting with a big number of different proteins, while others have only one or two partners. So this is a science called existing in nature. We do not need to create it artificially. It's self-assembling of proteins in the viral capsid. There is no genetic program for formation of such a nice structure. It's transmission electron microscopy. It's properties of the elements which bring together these proteins. And other examples, now it's artificial. It's self-assembly of lipids, proteins, cyclodextrin with surfactant, so layers, cubes, balls, they can appear as a self-organization of different elements. And these pictures were taken from the work of a German scientist from his group. It's Andre Groscher. And again, this is self-assembling topography. So he observed such different types of self-assembling of specific blocks of polymeric structures which appeared when they are brought together, solution, then evaporation, then using surfactants, etc. So other examples, you can see these polymers have different blocks. It's a 3-block polymer, and due to such specific structure in solvent 1, different structures can appear. This is already some sort of artificial intelligence, which is also in a focus of our workflow. So we can talk about an interface, which is a device or system that unrelated entities used to interact. And there are many publications on that regard, and I would like only to see the last paragraph when the composition of different elements together, so then we can use computation modeling and artificial intelligence to see the structure which will appear. And this slide, I took it during the conference in Washington 3 years ago, 4 years ago. So we have different generations of drug. This is generation number 1, which is used right now. We have liposomes, and it's possible targeting, not specific, just a liposome incorporates some medicine, and it can be also mRNA for anti-COVID. And second generation, we are already using some of them when it's already biomolecular targeting is used, when the drug has a platform, and on this platform there are different vectors which assist recognizing specific cells And finally, the third generation, it still does not exist. It exists only in the projects, in the research, in the papers. When there is a combinatorial nanotech, so these specific nanostructures are organized only on the place of its action. So the action is very focused there. So number three, how the multifunctional nanoscale carriers makes the medicine smart. In this part, a little bit theory. So the major shortcomings of the traditional anti-cancer chemotherapies. Number one is non-addressed action in the treated organ. Doxorubicin, a perfect drug, a very strong drug, but not you, but the patient can die in a week or in a couple of days of infarction, cardiac infarction, and because it possesses very strong cardiotoxicity. Then poor water, poor biocompatibility is related. Low water solubility of many drugs, and rapid development of to resolve these issues in our research. I will say about that a little bit later. So but it's development of the problem number one, non-addressed action. It was calculated that only 0.01% of drugs applied intravenously reach their real biological targets. 99.99% are useless, and when they are toxic, they are not only useless, but they are bad for the organism. Negative side effects, we'll talk about that later. And during a year, more than 50% of cancer and also tuberculosis patients gain resistance to applied chemotherapy. So this is an example how intravenous anti-cancer drug focused on brain tumor is distributed in the organism, in the body of the mice. 10 minutes. This is 99.99%. The organ does not need this. It needs only here, in the brain tumor. Then in 9 hours, it's kidney and more. All the rest is not needed and it's bad for the organs. So to overcome this problem, several drugs have been developed, and some of them they are already on the market. I already talked about the problems with doxorubicin, cardionephrohepatotoxicity, and myoset is doxorubicin encapsulated in the liposome. It produces enzyme pharmaceuticals. And doxil is doxorubicin encapsulated in pegylated liposome, produce ransom and ransom. And why it's better? Because the clearance from the organ is 600 times slower than of the doxorubicin, free doxorubicin. And it's concentrated effectively in blood vessels and tumors. And what is very important, possess 10 times less cardiotoxicity. So, why is this problem? The problem is because we have a plasma membrane, and the drug should cross the plasma membrane. It's the same in all cells. So 50% is blue balls are phospholipids, 50% are proteins, and some of them are receptor proteins, and there are also glycoconjugates, these yellow So, sorry, I should stop with the ring. So, fishes which are very non-specific or receptor proteins, or like widely used. These are antibodies now for biomarkers on the surface of pathological cells. These are cytokines. Some tumor and some inflammation cells have receptors for some cytokines. These are folate tumor cells have overexpressed folate receptor, salaminin, tumor-specific form. And these are monoclonal antibodies to transferrin receptors, and transferrin is on the endothelial cells, so it's used for crossing, for example, blood-brain barrier or blood tumor barrier. So, developing this issue, the hub idea have appeared. So first hub, it's like in the airport. First airport is to cross the endothelial barrier if the drug is injected intravenously. And the second half is to find the specific cell which should be targeted. And in both situations, the antigens are recognized by specific antibodies. So how it looks like, it's a blood vessel, and it's transcytosis using transferrin receptors. This small platform with the medicine gets inside, gets in tissue, and then it finds tumor cell using, for example, using HER2 receptor. So a new term appeared. It's a drug vectorization. So it's a drug with different vectors to find specific hubs. And usually vectorized is for this vectorization using polymers or metals or lipids. So during vectorization, it is possible to simultaneously deliver several drug substances which possess either opposite or synergetic mechanism of action. Right here, examples, applications. Multifunctional nanocarriers for simultaneous encapsulation of hydrophobic and hydrophilic drugs. Hydrophobic, many anti-cancer drugs are hydrophobic. Hydrophilic, for example, siRNA is for some peptides for membrane crossing. Another drug vectorization to modulate TCO distribution of molecular compounds, and again for vectorization of hydrophobic drugs. So I will present two examples of the multifunctional platforms for drug delivery. This is polymer dendrimeric structure, and you can see the drug, the folic acid, polyvalent glycol, antibodies, fluorescent dye for labeling, peptides for crossing the membrane, and MRM contrasting agents. So all this is intellectual of the platform. And another example, almost all the same, but linear structure, and the platform is polymalic acid. So on this polymalic acid, you can have monoclonal antibodies against the receptor, the growth factor receptor, monoclonal antibodies against transferrin receptor, crossing BBB, antisense oligonucleotides, again to mRNA for F2 and the epidermal growth factor receptor. mRNA contrasting agent rare earth metal, and fluorescent dye for bioimaging. So now what we are doing in my department. So here is the list of the drug delivery platforms we are using. It's unbelievable, but there are 6 of them. You can see polymeric electrolytes, Technical University in Wien, polyaccessory University in Germany, Tularen Criminal Technical University. The superparamagnetic iron oxide temperature sensitive particles, Institute of Macromolecular Chemistry in Prague, nature of zeolite dot with silver and rare earth metal cations, the Wien National University. And Hitosan we are preparing by ourselves, but this is very specific size of the Hitosan, and very specific proteins amination would be regulated. So size and amination of these particles from Hitosan are regulated. So why is that? We were not looking for so many nanomaterials. People were looking for us because we are good in bioevaluation. So they are coming, chemists, physicists, okay, we have such a platform, can you test it, how it works, how it delivers drugs, and etc. etc. So, typical story. Design and structure of nanoparticles for biology and medicine. That's what for young scientists, if they decide to do something, what they should take into account. They should take into account firstly size, which is important for clearance, not more than 100. Core, which defines the potential for biodegradability. Coating, usually polymer, defines the biocompatibility. Labeling, important for detection and bioimaging. Activations of this coating should have chemical groups for attachment of functional element. And biofunctionalization provides a possibility for particle recognition by specific cells. So, a few words about the amphiphilic polymeric carriers for drug delivery, which are developed by my colleague in Alexander Zaitsev from the National Technical University. It has hydrophobic part, it has hydrophilic part, and it forms its structure. So, Toxicity, which I told you, it's a phospholipid vesicle, liposome, with polyethylene glycol used for blocking non-specific elimination by the immune cells, and medicine toxicity. This platform has a polymer with phospholipid, polyethylene glycol, and toxicity inside and the same size. So I will go back to the main characteristics of the nanomaterials which should be taken into account. So if this is the property of your particles, you are the winner. If not, so first enhance biological action. So delivery in the nano form should improve the action, enhance the action. Otherwise, but other properties could be also important. So here it's cytotoxicity of doxorubicin and encapsulated form in human lung carcinoma cells and using the polymer published in German-Ukrainian nanotechnology. It's doxorubicin itself, deep action, and doxorubicin and its inhibition of growth on the nanoblock. And this is a vivo story. Again, you have increase in the weight of tumor when saline is applied. The polymer itself does not affect. Doxorubicin slightly inhibits. Doxorubicin delivered on the platform is strong inhibition of the tumor growth. And this is survival. The same picture, the alpha-leucine are bearing mice. In 15 days they are dying. With doxorubicin, 50% of animals survive. And with doxorubicin on the polymeric platform, at least 40-60 days, the animals live. And this is what's happening inside the body. It's acidic lymphoma, so it's action of free doxorubicin, and this is the action of doxorubicin on the nanocarrier, and these are dying cells. So, property number 2, even if the action is comparable, but the drug is absolutely not soluble in water. You know, who works in this field, you know that mostly one uses the metal sulfoxide, which is absolutely uncomfortable for the organism. So you need to have some way to improve water solubility, and there is even a term, PVSAD, poor water soluble anti-cancer drugs. So to overcome these properties, you need surfactants or co-solvents, which usually have adverse side effects. So when is the drug classified as poorly soluble? When the highest dose is not soluble in 250 milliliters at different pH. So here are some experimental anti-cancer drugs. Just for example, you can see that it's difficult to weigh water solubility from such a chemical structure. So to overcome this solubility problem, we use amphiphilic polymers. And again, this is the structure of the polymers designed and synthesized at Technical University, hydrophobic amphiphilic. And this is what is happening for water-soluble drug. The doxorubicin is simply non-covalently incorporated, encapsulated, and this is what's happening for water-insoluble drugs. They are only in the organic medium, and the drug is encapsulated in this, inside this micelle, and the hydrophilic chains are faced to water, so absolutely water-insoluble drug gains water solubility because it's hidden in this capsule. Similar situation with polyoxazoline, the group of Robert Luxemburg at the University of Würzburg, Germany. Now he also moved to Helsinki University in Finland. So these are poly-2-oxazolines, different types, and they have also this amphiphilic but hydrophobic domain, and which gives the water-insoluble drug inside the cell, and this chains which provide the water solubility. So this was the antibiotic, anti-cancer, with anti-cancer action. It's water insoluble, but with the polyoxazolide, heating, drying. Finally, we got, not we, but when we got the lead cells. And what is very important, we are happy with that in such a form, such nano form, the medicine, this experimental anti-cancer drug, can be dried, because it's a big problem. If even you get solubility, but stability of such a system is very short. Okay, it can be a month, it can be weeks, because in water solution the molecule is destroyed. But if you lyophilize, you can keep it for a longer time. So next property, also very important, enhanced transportation of the drug to the target cell. Here you can see different types of cells, breast carcinoma and drug-resistant breast carcinoma, and another type, the origin. But anyway, in black it's doxorubicin and its fluorescence inside the cell. And doxorubicin on the delivered on the Nano platform, depending on the drug concentration. And for drug resistance, it's very important, almost not in Gulf, but on the nano carrier is inside. And here the same situation. And another drug, not only doxorubicin, together with our Austrian colleagues in Vienna Medical University Institute of Cancer, we were working with ruthenium. It's already clinical trial stage anti-cancer drug. It has ruthenium as an element in its chemical structure, and we were targeting drug resistance, the resistant cells, deletion of p53 gene, and you can see free drug encapsulated on nanopolymer, free drug, the nanopolymer, or drug resistant free drug, very small action and encapsulated. So this is a picture how delivery looks like in 2 hours, doxorubicin, doxorubicin. Very little is inside the cells, mostly is on the border of the cells. But the same picture for the drug delivery on the platform. You can see it's inside. It's red fluorescence of doxorubicin in the nucleus. And another picture just to demonstrate very complex labeling of cells. It's green actin filaments, blue is DAPI or chromatin. So doxorubicin, you cannot see red fluorescence in the nucleus, and doxorubicin on the nano platform, you can see its interference blue and red, its pink color in the nucleus. So next characteristic which you can using the Nano platform. It's a preservation of the rapid release. It's a citation. It's so-called enhanced permeability and retention effect, EPR. So what it says, the drug rapidly gets into the tumor, but also due to osmotic pressure, rapidly released from the tumor. When the drug is delivered from the nanoparticle, it's retained in the tumor for a longer time. So we were using this experimental drugs, and you can see in 10 minutes, it's mostly in liver and kidney, where if you look in plasma, blood plasma, and brain, and it's slowly decreasing its concentration. And it's not only the drug, the polymer is slightly degradable. You can see this is thin layer chromatography. You can see that 20% is a fragment of the polymer and 25% polyethylene glycol fragment, and the excess of reds it's up to 10%, and the urinal reds up to 20% of the polymer. So it's released from the oil. Increased drug stability. Again, this is ruthenium containing nanomedicine, and called KP 1019 in the internet, and in 24 hours it's oxidized and it loses its activity. When the KP 1019 is on the polymeric area, in 24 hours it's intact. Enhanced crossing the biological barriers. Already told you that crossing the barriers is very important, because in a year, even a shorter time, drug resistance develops, and drug, especially anti-cancer drug, is useless. So there are three main types of the barriers which block the entrance of xenobiotics in the organism. So it's an organismal level, it's blood-brain barrier, placenta fetus barrier, and some other barriers. It's a cellular level, it's a plasma membrane transporting system for multi-drug resistance, so-called MDR multi-drug resistant. There are specific transporters for that, preventing the action of the xenobiotics and drugs. And this is molecular level, which is also very important. Even if the drug is getting inside, its action can be blocked due to action of systems, antioxidant, anti-radical defense, and protection of genetic apparatus with P53 and DNA reparation systems. So, example of action of doxorubicin and KP 1019 anti-cancer drug on the wild type and tumor cells with expression of multi-drug resistant systems. So we calculated an enhancement coefficient and IC50 of the drug on the carrier, IC50 free drug, and we observed that up to 20 times enhancement of the action towards the drug resistance systems. Other lung cancer, other types of how it works. So this is the pumping system, BGP 200 of MRAP pumping system. Doxorubicin, for example, is getting inside and not getting to its targets in the nucleus. It's pumped of the cell because of the work of this drug resistance mechanism for overcoming drug resistance. But tumor cells are using this mechanism, and it's very important for them. And if you have the anti-cancer drug encapsulated, so this mechanism, this transporting system cannot see the drug. It's invisible for the drug. So it's pumped up. It's very important and decrease of negative side effects in vivo using drug encapsulated and in free form. You have a lecture in the morning regarding toxicity of different xenobiotics. So you can say that nanomedicines function like a double-edged sword. It's positive effect, enhancing treatment and diagnosis, and negative effects when the drug is possess general and specific toxicities in the organism. So for those who would like to know a little bit more, I do recommend to use the keywords pharmacokinetics, analyzing the status of the drug in the body, absorption, distribution, bioavailability, bioequivalence, metabolism, and expression, and pharmacodynamics, which characterize molecular responses to drug action in the body, molecular interactions. So these two disciplines, these two directions of study are very important to evaluate the negative side effects. So what we were studying and what other people also studied as a negative side effects. Hepatocardio, nephro, neuro, hormonal, geno, environmental toxicities. So we were studying all of these, not studying neurotoxicity. So how can we decrease the toxicity? It's let's say a simple way. If the substance is toxic, so you should use less concentration, but then it will stop working. So it's one way, but it's used in the nanocapsulation process. Or we can mask or hide the highly toxic drug in order to avoid its non-addressed action. So if the drug, and we demonstrated that, I have no time to show, that the effect of the drug could be achieved in 10 times less concentration, and then there will be no effect of high dose. In 10 times less concentration when it's delivered in the nano-capsulated form, comparing to delivery of free form of the drug. And we are studying different indicators of hepatotoxicity, enzymatic total protein, etc., of cardiotoxicity, different indicators, under one is creatine phosphokinase. Nephrotoxicity is cations, anions, total protein, urine, creatinine, glucose. And as an example, just pay attention. Stop short and its glutamine transferase, indicator number 1 for hepatotoxicity. So, some level is present in blood serum, but after doxorubicin action is increased very, very strongly. And when doxorubicin was applied with the polymeric nanocarrier, so it's like in the control. Similar for creatine phosphokinase as indicator of cardiotoxicity. Control increased in the reaction of doxorubicin and normalization when doxorubicin was delivered on the nanoplasma. There is very nice experiment. If the tissue is damaged, so some element should escape from such tissue to blood vessel. And there are different isozymes of lactate dehydrogenase in heart tissue and in blood. So here, this enzyme is originating from the heart and it's appearing at the action of doxorubicin and it's appearing in blood plasma. When the doxorubicin is used on the carrier, so there is no such a strong increase of this heart-specific enzyme in the plasma. And just we did nephrotoxicity here, control doxorubicin, and its strong decrease in total protein and normalization when doxorubicin was applied on the nanocarrier. And most of the other, like sodium, strong decrease, normalization. Glucose increase, not total, but normalization. So immunotoxicity, up to 5-6 years ago, maybe 7, it was not addressed very strongly, but now we address it also. So, immune system is dependent on the immune cells, and these are granulocytes, neutrophils, xenophils, basophils, monocytes, and lymphocytes, T and B cells. So what we found that lymphocytes decreased in blood serum of B16F melanoma-bearing mice. And when the anti-cancer drug was applied on the nano platform, there was a normalization. Neutrophils were increased, opposite, but using the nano medicine, it was normalized. Erythrocytes falling down, normalization in the action of the nano platform. It's a very fashionable to study neutrophil extracellular traps. When the neutrophils see something bad, it throws away the DNA. The DNA is easily stained with a cursed pyridium bromide. So here, positive controls neutrophils, it's a control neutrophils, throw away the DNA to trap these bad substances. And here, supermagnetic particles made and coated with the polymer and coated with is the silicate. You can see a big difference when these particles are coating with some shell. It's the same, but in calculation. So genotoxicity, I will not give you the examples, only name the methods which are used for genotoxicity studies. Ames test, it's using bacterial mutation in bacteria, salmonella, mammalian cells for detection of recombination. Anatelophases assay in onion cells for detection of chromosomal alterations. And comment assay for breaks in the strands of the DNA. They are using all of this. The environment toxicity. You know, there was a lecture about the Titan dioxide particles, and it's used everywhere, and due to that, it's appearing in the environment, and it's toxic for the living beings in the environment. So here we have a series of papers in collaboration with our colleagues from Ternopil Pedagogical University, and found different toxicity for bivalve mollusks, for carp, and for amphibia. So we found that mollusk is less toxic for metal cobalt containing nanocomposites comparing to fish, which can release this toxic element from these nanocomposites. So what is, and I'm close to finishing, just in time. So what is very important in your study, everybody's study, to have this therapeutic index as big as possible. So therapeutic dose-dependent effect and toxic dose-dependent. This toxic dose-dependence effect should be far to the right and therapeutic far to the left. Then this index will be okay. So new trends, this is just what you should take home after listening to this lecture. So I was talking about so many particles, but up to now there are very few FDA particles. Since 2 million papers, but only liposomal drugs, 5 liposomal drugs have been FDA approved, and 2 polymers have been used for particle preparation, anthraxane. It's a packed cell on the albumin, and it's cyclodextran based on nanoparticles. So you can realize that most of them are biodegradable and they are of natural action. That is why we are still developing this direction with chitosan, because 5, the rest, they are still not, the organism can be not very happy with them, but chitosan is absolutely non-toxic biocompatible. So, oops, sorry. Yes. Still the market says everything is good. So protein-based nanopharmaceutical products will double up to 2025. Nucleic acid-based will double, and we have these liposomes keeping the RNA, and it will develop very rapidly. And small molecule-based market will also double, and we have many anti-cancer drugs based on such a value. So this is mRNA vaccines, a new area in nanomaterials. So there are 22 mRNA-based and 15 DNA-based vaccine candidates for clinical trials, including 3 mRNA-related nanoparticle vaccines. So this field will develop very rapidly because of the technological situation. So nanotheranostics, just it exists. So this is combination of therapeutic effect and diagnostic effect. So it's rapidly developing field in the nanomedicine. Take home, true nanomaterials should possess such important characteristics: enhanced effectiveness, accelerated delivery, circumventing biological barriers, reduction of adverse side effects, prolongation of stability, providing of water solubility. If the nanomaterials you will develop possess all of this, you will be happy, and we cannot avoid these issues because still many medicines do not possess really adverse delivery, cannot cross the barriers, are poorly water soluble, so we should take that into account. So problems still exist. I already named all of them. I would like only to stop on the last. Low reproducibility of synthesis of drug carriers stops their certification as medicinal remedy for FDA. Polymers and other chemical structures. You cannot be 100% sure that from one synthesis to another synthesis you have the same action. So if there is something bad, you can say, okay, this sort of particles are bad because they were not reproduced in the synthesis. So just a slide again from the conference in Washington. Mauricio Ferrari is a great man in nanomedicine. So he was 4 years ago. He said that at that time he already used from different grants $111 million for developing nano platform based on silicium oxide. And still there is no drug. So he is developing. And many issues which I touched today have been included in the monograph and the editor of this monograph in the Nature. Biomedical nanomaterials from design and synthesis to imaging application and environmental impact. So, the battery is there. Can you move 2 slides for acknowledgement? Because the battery is not working. Talking for too long.
So,
I would like to thank the collaborators of my department, of course, and outside Ukraine partners, Institute for Cancer Research in Vienna, Medical University of Essen, University in Germany, University of Germany, even now. Sorry, I'm not naming the names of the professors. Technical University of Germany, Institute of Macromolecular Chemistry, these are magnetic particles in Czech Republic, Citizen Medical Center, supportive some visiting conferences in USA and in Ukraine, BTech, National Technical University, National University Institute of Biochemistry, Institute of Biochemistry, National University, Medical University. By the way, this is entrance to the Bill Palace of Sciences. Next meeting, you should organize here in this fantastic building. For those who were there, who saw this building, I think you will confirm. So these people are outlines keeping our collaboration. Thanks for your attention, and if you have questions, we'll be happy to answer.
Thanks a lot. That was really broadly covering, and I think it was also very, very well presented. So, please.
So I have a question. Which type?
Loud, or come down.
Which kind of particles and for what types of cancer are used for cancer treatment in Ukraine? If there are, because you're naming some of those drugs and nanoparticles.
So, I named in vitro systems, in vivo nanomedicines used because they're very expensive. Even Doxil, years course for treatment of cancer with Doxil, it's more than $10,000 US, and of course it cannot be for everybody because the patient should pay for that. So, and again, 5 slides to the end, there are only very few nanomedicines FDA approved, and we should take into account FDA recommendations. Otherwise, we'll be experimenting on people, and it's forbidden.
Okay, thank you. And one more, you said that there's a real low percent of diagnostics
So, please speak up.
I understand the question.
And is there a way to make standard those?
Good question, and this is mostly for the polymers. If you use albumin as a carrier, you will not have such a problem. If you use other natural products, you will not have such a problem. But for chemical polymers, you will have it always, and probably you will have less problems like this for inorganic particles, because it's easier to normalize to standardize their characteristics. But mineral particles, up to now, there are no FDA approved nanomedicine based on mineral particles. It's only organic origin particles.
So, we have time for a few more questions, but then it would be really nice if you go up there, because then we have you in the video screen. So, first, more questions. Yeah, Oksana.
Yeah. Did you try ketosol nanoparticles for in vivo experiments?
Please come loud.
Sorry.
Yes.
Did you try ketosol nanoparticles for in vivo experiments? Because I have tried to combine with methylene blue, and it can block vessels. Did you try it for with mouse?
Yeah, the blood vessel problem is really a problem because firstly, the drug should be released from the vessel, but there could be different aggregations, and very frequently such aggregations of the materials happen, and people say, okay, we don't want to use such a nanomedicine which blocks the vessels.
Okay, thank you. So, more questions. Well, maybe then during the next break or in the evening.
I'll be available tomorrow and today to the end of the working day.
And it would be really nice if you join us, as hopefully everybody, for Science at the Bar in the evening. It turned out last Tuesday that it is a really nice event, and today we have a second service. So you are, of course, also invited to the bar, which is quite close here.
Thank you very much.
Okay, thanks again. Was this So you stop the live stream now?
Yeah.
Okay. Okay, so I do the connection to the next talk.